Aging is an intricate, multifactorial process involving dynamic interactions across all levels of biological organization, from DNA to systemic physiology. For decades, researchers have attempted to define the biological underpinnings of aging using reductionist approaches, focusing on single genes, proteins, or pathways. While such efforts have yielded crucial insights, they fall short of capturing the full complexity of human aging and the determinants of exceptional longevity. In recent years, a shift toward multiomics, the integrated analysis of genomics, epigenomics, transcriptomics, proteomics, and metabolomics, has provided a powerful systems biology framework for decoding the biology of aging in its full multidimensional context. Genomics, the study of DNA sequence variation, forms the foundational layer in multiomics aging research. Longevity-associated genes such as FOXO3A, APO, and SIRT6 have been identified through genome-wide association studies, GWAS, particularly in centenarians and long-lived families. These variants often confer resistance to age-related diseases by modulating stress resistance, metabolic regulation, or DNA repair. Yet while genetics sets the blueprint, it only partially explains why some individuals live significantly longer and healthier lives. Heritability estimates for longevity hover around 20 to 30%, suggesting that additional regulatory layers must account for the rest. Epigenomics provides the next layer of insight by examining chemical modifications to DNA and histones that regulate gene expression without altering the genetic code itself. Among the most promising tools in aging biology are epigenetic clocks, such as those based on DNA methylation patterns, which can accurately estimate biological age and predict morbidity and mortality. These clocks not only provide biomarkers for aging research, but also illuminate how environmental exposures, lifestyle, and disease interact with inherited genomic architecture. Importantly, long-lived individuals often exhibit younger, epigenetic ages than their chronological peers, suggesting slowed or buffered epigenetic drift over time. Transcriptomics, the study of RNA transcripts produced by the genome, adds a dynamic view of gene activity across tissues and time. Aging alters the transcriptome in tissue-specific ways, affecting pathways related to inflammation, proteostasis, and mitochondrial function. For example, aged tissues frequently show increased expression of pro-inflammatory genes, a phenomenon linked to inflammaging. However, transcriptomic signatures in long-lived individuals often maintain patterns consistent with youthful gene expression, particularly in genes related to stress response, DNA repair, and metabolic stability. These signatures may reflect both inherited and environmentally reinforced transcriptional robustness. Proteomics extends this analysis to the actual proteins that execute cellular functions. Protein abundance, structure, and post-translational modifications shift dramatically with age, influencing key pathways such as autophagy, immune function, and synaptic signaling. Aging is often associated with a decline in proteostasis, the cellular capacity to maintain a balanced and functional proteome. Proteomic analyses in centenarians and their offspring frequently show preserved expression of proteins involved in antioxidant defense, chaperone-mediated folding, and metabolic regulation, factors that contribute to cellular resilience. Metabolomics, the study of small molecule metabolites such as lipids, amino acids, and signaling molecules, provides a real-time snapshot of biochemical activity. As the interface between genotype, environment, and phenotype, the metabolome is highly sensitive to aging and lifestyle. Age-related changes include altered lipid profiles, accumulation of oxidative byproducts, and shifts in energy metabolism. Notably, long-lived individuals exhibit distinct metabolomic signatures characterized by low insulin resistance, favorable lipid ratios, e.g., low LDL/HDL, and enhanced antioxidant capacity. These profiles often mirror those seen in caloric restriction studies and other longevity-promoting interventions. When combined, these omic layers allow for integrative modeling of aging as a complex, dynamic network. Multiomics approaches have been instrumental in identifying molecular signatures of healthy aging and health span. For instance, systems-level analyses of centenarian cohorts have revealed coordinated regulation of immune response genes, metabolic stability, and preserved mitochondrial function. Machine learning algorithms applied to multiomics datasets are now capable of predicting biological age more accurately than any single omic measure alone, offering promise for personalized aging diagnostics and interventions. Moreover, The systems biology view afforded by multiomics is beginning to reshape our understanding of aging not as a single process, but as a mosaic of interdependent trajectories, some of which may be modifiable. For example, individuals may exhibit accelerated biological aging in one domain, e.g., metabolic or immune, but not in others. By dissecting these trajectories, researchers can begin to identify specific intervention points, whether pharmacological, dietary, or behavioral, that target aging with precision. Emerging applications of multiomics include the development of composite health span biomarkers, longitudinal monitoring of aging trajectories, and drug discovery platforms that identify geroprotective compounds based on their ability to revert multiple omic signatures toward a youthful state. In this regard, interventions such as metformin, rapamycin, and senolytics are being evaluated not only for their clinical effects but for their capacity to shift integrated omics profiles. Another frontier involves the application of single-cell multiomics, allowing researchers to map how aging unfolds at the level of individual cells. This is particularly relevant for tissues like the brain, immune system, and skeletal muscle, where cellular heterogeneity plays a critical role in function and decline. Single-cell technologies can unravel how epigenetic silencing, transcriptional noise, and proteomic remodeling contribute to senescence, regeneration failure, or disease vulnerability in aging tissues. In conclusion, multiomics approaches offer an unprecedented window into the biology of human aging. By integrating diverse molecular layers, researchers can move beyond the limits of single-dimension studies and toward a more holistic, actionable understanding of longevity. While still in its early stages, this systems-level framework promises not only to clarify why some people live longer and healthier lives, but also to guide interventions that optimize aging trajectories for the broader population. The challenge ahead lies not in generating more data, but in weaving it together into coherent models that inform both science and clinical practice in the era of precision aging.